Sound, teach them skills about writing and analyzing texts using that particular topic as a central point. Today, I'm delighted to introduce to everyone Dr. Seth Bordenstein of Vanderbilt University, who is the high priest of the Holobiome. And what I have done, as in previous talks, is given the students a series of articles to read. They sent me questions. I sent them to Dr. Bordenstein, who is here today. And with that, I would like to introduce the class to Dr. Bordenstein and Dr. Bordenstein to the class. Here you go, folks. Take it away, Seth.
Okay, so first of all, I'm jealous that I'm not snacking there with you right now. That looks very good. Second of all, I'm really happy to join you guys today for a number of reasons. One of which is science isn't just for scientists. And so the outreach that we do with your classroom, the outreach we do beyond our labs is probably the most important thing we do, because if we just keep the science to ourselves, then what good is it? Science is no good unless it's shared, either as fundamental knowledge or as some application. And I think the topic of symbiosis, in fact, used to be a topic of mostly just fundamental basic science. And that has dramatically shifted in a way that's profound for our field, because now symbiosis is being looked at as a fundamental science, essentially fundamental to all of the life sciences, as well as applications are coming online quickly and will continue to come online, I think, in the next 5 years. For example, I was just reading a New York Times article today about the company Microbiome making a pill for fecal transplants that's basically, instead of getting a fecal transplant to cure a particular illness, they'll put the fecal stuff into a pill, and the person takes about 30 pills, and in one instance could be cured of a gut infection like Clostridium difficile. And so these kinds of interspecies interactions that you guys talk about and that are being applied today, and that example is just a dramatic shift from where the field has been since I've been studying it for the last 20 years or so. Okay, so the other reason I'm happy about joining you today is because I want to teach a course a lot like what you guys are doing right now. Traditionally, I teach a lecture course called Microbiology, and I also teach a seminar course for upper-level juniors and seniors on contemporary literature in symbiosis. We call it Living in Symbiosis. And it just kind of struck me over the last year or two that maybe we're doing this the wrong way, and maybe you guys are doing it right, in the sense that if we really want to get people inspired about science that affects their college pursuits, or if we just want to get scientific literacy into the course room or into the college at an earlier level than just sitting in lecture classes, then doing this at your level is probably the better way to do it. And one of the things that I'd like to do, and maybe Doc Martin will do, is actually track your success to some extent. You know, what I'd like to learn from our students here at Vanderbilt in Nashville, you know, to what extent do they stay in science if they've gone through a course like this early on? Or to what extent do they delve into scientific literacy even if they're not in science as they move on in their careers or in their tenure at college? So for those general reasons, I'm excited to join you today. And for more specific reasons, I've taken a look at your questions, and I'm happy to start answering them. Okay, so let me move some windows in my background here. Genevieve, which one is Genevieve? Who's Genevieve? Hey there, can barely see you. Okay. All right, so I liked your question to start off with because it's pretty straightforward. You know, what got me interested in the microbiome? Do you think this will change people's perspectives on individuals and how they operate, especially in the medical world? Is that your question, Genevieve?
Yes.
Okay, great. So feel free to chat with me if anything, you guys want to bring anything up. But I think getting interested in the microbiome is somewhat of an interesting question because there was symbiosis before there was microbiome. And that's all that we're essentially talking about. And so many pioneers in the field have been studying the microbiome under a different lexicon, if you will, symbiosis, for decades. And the story is similar for me. I started in graduate school in 1997. And in fact, I started in undergraduate years doing research on the microbiome, if you will. And I was interested in studying one particular bacteria in the microbiome of insects called Wolbachia. And Wolbachia is sort of a science fiction story come true, because this bacteria lives in the gonads of arthropods or insect hosts. We're talking about testes where sperm are made and eggs, or ovaries where eggs are made. These are the places where the bacteria hang out. We don't often talk about bacteria in the reproductive system and how that might affect the way individuals reproduce. But in fact, when I got into the Wolbachia field, it was pretty clear that these Wolbachia bacteria change dramatically the way a female will reproduce. For example, it can change the number of female offspring versus male offspring, literally turning a mother who gives about 50% males and 50% females into a mother who lays 100% females. And so it's essentially a way of feminizing, if you will, the population or the species to a degree that just seemed like science fiction at the time it was discovered. Now, there's a good answer for that, and it's a good moral of the science story here, is why would Wolbachia create these gender-bending capacities to make more females in the population? Well, I was fascinated by that. In fact, the Wolbachia do this because they're exclusively transmitted from mothers, not fathers, mothers to offspring. So as a result, if you're only transmitted through females or your mothers, then from an evolutionary perspective, males are a dead end, not useful. Might as well run around them, kill them, do whatever you can, because if you make more females in the population, you make more of the transmitting gender for Wolbachia. And this kind of blew my mind as an undergraduate, and it still does today. And I think one of the hottest questions in the field right now is, how do Wolbachia do this? What are the genetic capacities, if you will, of this bacteria to distort host animal reproduction in sometimes very complex ways, if you think about it, that a simple bacteria has to do this dramatic alteration to host reproduction. So we have been studying that for 20 years in various ways. And so I would consider that one member of the microbiome, perhaps one of the most interesting members of the microbiome. And my PhD thesis, so from 1997 to 2002, was focused in on how these Wolbachia bacteria fundamentally drive the origin of species, new species. And this is really what piqued my interest as a student and as a budding scientist, because I was intrigued by Wolbachia and its gender-bending capacities. And then I was also intrigued by a course I took in evolutionary biology as a sophomore. That's what led me to go do research. And in fact, it was the first time in my academic career that I realized I just didn't want to do biology for the sake of biology or becoming a pre-med student, for example, but I actually wanted to delve much deeper into this curiosity, the origin of species. And so that's what I did my initial work on for my PhD. We were looking at how Wolbachia can essentially cause the wedge that splits two populations into good species. And this is a question that, you know, has been on the minds of biologists since Charles Darwin wrote The Origin of Species. He wrote The Origin of Species in 1859. I advise all of you to at least have a copy or read some sections of that book, because the writing is complex but exquisite, and the writing is beautiful in the sense that it takes on arguments and counter-arguments and critiques all at the same time to pull together a synthesis. Anyways, if you march forward into the 1920s and 30s, DNA was the hot topic of the time, finding that DNA is the basis of hereditary material, and ultimately finding the mathematical theories that would ultimately merge with Darwin's thesis on the origin of species, centered biology squarely on nuclear genes as the agents, and even I would go as far as to say at that time sole agents of speciation. So symbionts and microbes were just not part of the lexicon, and they really weren't part of the lexicon until the 1970s, where we actually got a breakthrough from Lee Ehrman's lab. And then things rayed pretty dormantly till the 1990s, which is where myself and other labs started to come on board to the idea that bacteria and microbes could be an essential part of speciation, at least for some insects, if not many types of animals and plants. And what happened was we found that Wolbachia caused this sperm-egg incompatibility, where the sperm from one species get modified by Wolbachia, and when they fertilize the egg of another species, that modification doesn't allow the sperm and egg, actually the sperm and egg fuse, but it doesn't allow that egg to develop properly. The sperm modification is actually a paternal genome or a male genome modification, and the egg essentially dies when you cross two species together that have been modified by Wolbachia. But surprisingly, if you remove Wolbachia from the host by antibiotic, just getting rid of an infection, you can cross the species again, and now the sperm and egg, without the modifications, fertilize and develop absolutely perfectly. And so the primary thing isolating these particular insect species that we were studying were the Wolbachia bacteria that just love to mess around with reproduction. So that's what put me on the path to study the microbiome, and ultimately shifted from studying one bacteria to studying the whole community of bacteria and animals, because I became somewhat satisfied that Wolbachia, the evidence suggested that Wolbachia was a speciation agent, not only in the systems that we were studying, but in other systems. But there was always this critique coming in the background that it was just one bacteria that could potentially cause speciation. And so what are the ways in which we could broaden the field of symbiosis in the microbiome to essentially a universal aspect of speciation? And so around 2006 or 7, I was at the Marine Biological Laboratory in Woods Hole, Massachusetts. I was next to the lab that developed the sequencing technology that has led to the explosion of microbiome research, namely 16S ribosomal RNA gene sequencing. And at that point, it occurred to me that we could plug that technology in, not just from sort of environmental surveys of microbial diversity, but to host-associated microbial communities. And we could look at how these host-associated microbial communities potentially affect speciation. So that's kind of a long-winded answer, but one that's worth setting the framework here for sort of how I got into science at an age that you guys are about right now, taking that course that I loved, finding a connection to that course, and then a further connection to symbiosis, which broadened the scope of what I do now. So the follow-up question is do we think people's perspectives on individuals will change, especially in the medical world? What we're referring to here is the article and the topic of holobionts. And so holobionts is a term that refers to the host plus all of its associated microbes. And those microbes might be transient, or they might be permanent, stable members. Anything goes. It's just a very general term. And the short answer is yes, I think this will change the medical approach and thinking. But first, let me say that the word holobiont or hologenome is absolutely essential right now because, in my opinion, because we've had sort of loose words like superorganism to describe the complex of you and your microbes, or you might have heard of calling your microbes a second organ, if you will. And these terms, superorganism or organ, are actually intended for other things. So the superorganism refers to one species that socially interacts like a eusocial ant colony that operates as one larger organism. But that's just one species. It's not the microbiome and its host made up of multiple species. And same goes for the organ. The organ is made up of cells from one genome, right? And that genome encodes the cells that make that organ. But the microbiome is, of course, a consortia of complexity in cell types and species and genomes. So we really haven't had a proper term for holobionts until, actually that term came up in 1990s by Lynn Margulis, a very famous symbiosis luminary, but really was resurrected lightly in papers, but didn't really get the attention I think it deserves till more recently with the explosion of the microbiome field. So what does this mean for medical studies? It means that when we do medicine, or when we treat somebody and we want to do precision medicine or personalized genomics, you know, the president launched the Precision Medicine Initiative at a State of the Union address last year. And he talked about having personal genomics be part of the norm in medical care and preventative care. And what he meant by that is, let's sequence human genomes as a way to understand the basis of diseases. But as you're learning, hopefully, there are only 20,000 genes in the genome, and there are up to 10 million genes in your microbiome. And we've had a very difficult time sometimes linking disease causes to genes of major effect in your genome. And I think what we'll find is that some of these diseases that are difficult to track down in your nuclear genes may have large influences in your microbiome. And one good case of that is obesity. For example, we can, based on your microbial signatures in your gut, we can predict obesity with a very high accuracy, almost 90% accuracy. And you can't do the same based on your nuclear genetics. So in that case, your microbes tell you more about a particular state or physiological condition or illness than your genome. And actually, the holobiont is all this stuff together. The hologenome is all of the genes from the holobiont stuck together. And we need to appreciate that as a unit as we consider what makes us healthy, sick, and how organisms adapt and evolve in nature. Okay, so let me move on. Gloria, can you raise your hand, Gloria? Hi there. Okay. Even though there are many different organisms in a microbiome, can they vary from person to person? So there are many specific to an individual, still making them I and separate from the rest. So they do vary from person to person, but I think this is an interesting, nuanced question, because you often hear that each of us has our own unique microbial signature, right? That you sitting next to your peers right there may have 90% of your microbes different from your peers. And that's technically accurate based on how we define a microbial species or taxon. But that definition could change, because at a very micro level, you may have microbial species that are unique to you. But at a general level, we actually all share similar types of microbes. So if you go down the taxonomic order, kingdom, phylum, class, order, family, genus, species, if you look at the phylum level of all the bacteria that are in your guts, it's pretty much going to be roughly the same. We all have Firmicutes, we all have Bacteroidetes. So how individualized are we if we classify taxa at the phylum level? Not very much. How individualized are we if we classify bacterial taxa at the species or strain level? A whole lot. But what is a bacterial species? I don't know if have you guys crossed that landscape yet?
No, but I'm dealing with that right now in my microbiology class. It's a fun question.
All right. So we tend to have a good idea of biological species being things that don't interbreed. Animals and plants tend to obey these rules, give or take some details. Microbes just don't obey these rules at all. Right? They exchange genes randomly between each other. So we tend to define microbial species by sequence identity. That's very much an arbitrary, non-biological, non-phenotypic perspective of a species. So whether we share lots with each other really depends on that percent similarity cutoff in their DNA to determine whether you share a lot of that same microbial species or DNA or not. Okay? All right. Colin,
Colin is ill today.
Okay.
His microbiome is acting up.
Okay. I will move on then to JT. Hey there, JT. All right. How are you liking this class so far, JT?
I love it.
Yeah? What do you like about it?
It's really interesting to me, especially learning about the symbiosis, like the microbiome and everything within us. Really interesting because I'm interested in going into the medical field. So it's interesting to see how this kind of emerging and new hot topic in biology can influence people so greatly.
Was this a new topic to you?
Yes.
And do you feel like it'll affect the way you further study in college?
Yeah, I think so. That's actually what I'm writing my term paper on.
Yeah, okay, great.
What's the topic you're doing, JT?
The brain-gut connection.
Awesome. Fascinating area. I think just as we talked about fecal transplants or poop pills coming online, we're going to see pills that do things for the gut-brain axis as well. So people are calling these psychobiotics, as you may have come across already. The idea is that you'd be taking a probiotic to affect your anxiety levels and beyond. And so it's not my exact field of expertise, but it is a tremendous, tremendous new world to think about, because we've literally never thought about neurobiology beyond the brain and the nervous system. But in fact, we're now learning that the gut microbiota is central or influential, at least, to the brain and the nervous system.
Seth, I wanted to say that I made them purchase Rob Knight's short book based on his TED Talk, and we're having Dan McDonald as our last speaker in this group. So, you know, they get the highs and the lows. I've already told them if you want an area sequenced, you just whisper it into the night, and the next day Rob Knight sends you the FASTA files. Am I wrong?
Fantastic. Yes, he might be a little busier now than he used to be, but we'll get him back. Yeah. Okay, so JT, you asked, the review article mentions that you or I said that the silos of microbiology, zoology, and botany are breaking down. How do you think they are deteriorating? And how do you think advances in this new biological approach will help them? So they're deteriorating because there is less of a disciplinary boundary in the way we do our science when we study an animal or when we study a plant. Because it's becoming apparent that if you are a zoologist or you are a botanist, that most of the phenotypes you're studying are affected by the microbes associated with that animal or plant. And so there's no reason to separate the biology of the organism out like that when we now appreciate it's so interconnected at a functional level. And so that's how, at least I think most people mean when they feel or when they express that these silos are a little outdated and that they're breaking down to fuse. And the concepts of holobionts and hologenomes is really a lexicon to reflect the merger of these disciplines. And, you know, it's an idea that we're just starting to get out there. I think as biology continues to go forward, and as we see the integration of, let's say, a Mendelian genetics approach, somebody who studies genetics only, just what happens in the nuclear genome, those investigators will realize that perhaps the phenotypes they're trying to link to the nuclear genome can only be fully understood, or in fact more understood, by taking into account the microbes that also associate with that organism and in fact interact with the genes in the nuclear genome. And so one could also argue this vice versa. If you're just a bacteriologist or a virologist who happens to study a host animal or plant because you want to know how that bacteria infects that host plant or animal, we can't do that in a bottleneck of just looking at the biology through the lens of the microbe. So one could ask, what are the genes that the microbe uses to colonize that host? What are the virulence genes, mechanisms? Could I knock out those genes and prevent virulence? But you know what? That microbe is colonizing a susceptible host, right? And that host is susceptible because it has some sort of genetic repertoire or physiological condition from that genetic repertoire that makes it susceptible to that particular microbe. And so we can't just understand what makes a bacteria virulent without understanding what's happening on the host side, and vice versa. We can't understand a particular host phenotype and the genes or mutations that affect that phenotype outside of understanding what are the microbes doing to affect that phenotype. So that's the important point about fields breaking down, and that, you know, in 10 years from now, when we have a new cohort of biologists going into graduate school, or you guys could be going to graduate school and leaving at that time, how will departments look, right? Will we have departments of zoology and botany and microbiology? Perhaps, because change in science is very slow, and rightfully so. But also, you know, maybe the norm will just be large departments of biological sciences. You know, no longer will we have universities with 11 different life science departments. Maybe we'll have a school of life sciences with subdivisions, but there has to be greater crosstalk. There has to be an exchange of paradigms, and there has to be a way to communicate this kind of thinking, which is interdisciplinary by definition, across the lexicons, individual lexicons of all those disciplines. Because we all sort of in science have our own ways to categorize things within zoology or within botany or within microbiology, but perhaps there's another language that's more synergistic that we have to start thinking about using. And perhaps these are some of the words we'll end up using more commonly. Okay, Caroline. Hey there, Caroline. So what was your major coming into the university this year? Or what did you think you want to major in?
Biology.
Just biology in general? Do you have a specific interest, or is it general biology?
Marine biology.
Tell him why you like marine biology.
Because of Riftia.
She loves Riftia.
Awesome. Okay.
And someone you know will be talking about Riftia shortly, Irene Newton.
That's right. Exactly.
There's a connection there, isn't there, Seth?
Yeah, it's one that I'm not going to step on right now. So you guys will have a great conversation talking to each other. So Caroline, you asked, what is a phylosymbiotic signal and how does it work? And phylosymbiosis is akin to phylogenomics. Have you heard of the word phylogenomics? Have you heard of the word phylogeny?
Yeah.
Maybe? Okay. So let me tell you what a phylogeny is. A phylogeny is a tree-like structure that depicts the relationships of organisms to each other, right? So two organisms on the tree structure that are more closely related to each other will be also visually closer to each other on that tree structure. And you can go down the list, increasing divergence across the branches. So when we make phylogenies, we typically use gene sequences today, and we're comparing how similar two organisms are to each other, to a third, to a fourth. Now, phylogenomics, before we get to phylosymbiosis, popularized by Jonathan Eisen, who's a well-known biologist at UC Davis. Phylogenomics is basically using not just one gene to make a phylogeny, but all of the genes in the genome to make what may be a more accurate phylogeny, because each gene has its own individual history of evolution, and forces acting on that gene might be different than the forces acting on other genes. So if you make a phylogeny of relationships of organisms based on one gene, it could potentially be a little different than another gene. So the phylogenomics approaches take the whole genome and now make a potentially more robust phylogeny. Phylosymbiosis follows that now because essentially we're taking microbial communities, that is all of the species associated with a particular host, and asking, how do microbial communities relate to each other? If this is the phylogenomic or phylogeny of a particular animal or a plant, what would the relationships of their microbial communities look like? And if they parallel each other, if the microbial community relationships look like the host tree, that is, the microbe tree looks like the host tree, there's concordance, and we call that phylosymbiosis. And so it's essentially a way of going from fingerprinting ancestry through genetics in the genome to now using microbial community information to do the same thing to potentially test for whether there's a signal of ancestry in the microbiome. So we've applied this in our parasitic wasp system. We study these tiny little jewel wasps. They're shiny and metallic. And we have 4 close related species that all diverged in the last couple 100,000 years. And we know their relationships based on their host genetics. And when we sequenced their gut microbiomes and compiled their relationships, we also found that there was a phylosymbiotic pattern. And to us, this tells us that there's a sort of ancestral fingerprint in the gut microbiome as much as there is in the nuclear genome. And that there's the potential for parallel divergence. That is, the genome and the microbiome are diverging in parallel to give us these phylosymbiotic patterns. Is that clear? Okay. Any other questions on that from the class? Good. Okay. So phylosymbiosis is kind of spreading around. This has not only been observed in Nasonia, but it's also been observed in Hydra. It's been observed in chimps and other hominid species. It's been observed in ants as well. And we'll see how far this pattern goes on whether there's a more universal signal of ancestry beyond the genome in the microbiome, further tethering the stories together of the microbiome and the genome. Okay. Christopher, where's Christopher? Well, hello there, you're off the screen a little bit. Mark, can you move it over there?
Sure.
Thank you.
Wave again, Chris.
I
All right, hey there. So how did you decide to take this course on symbiosis?
Well, it was definitely looked like the most interesting of the seminar classes, I'm gonna be honest. And I really enjoy biology and kind of like the weird part about parasitism. And I always thought that symbiosis was always a very interesting connection between different organisms.
Great.
What's your term paper topic, Chris?
Mine's on horsehair worms. So it's
On what?
horsehair worms. They're pretty fun little organisms. They make, well, it's up for debate still, but they make their host commit suicide, and then they exit through the lower end of their host, and they're typically 3 or 4 times longer than the host they were inside of. So it's pretty fun stuff.
Yeah, amazing. Don't get any smart ideas from that. So how long is your term paper? How long does it have to be?
It's 3 to 4 times his height.
Right now, I think mine's close to 10 pages,
Yeah.
but it's kind of, there's a lot of stuff I could have put in. So right now, it's kind of figuring out what is important and what's just being redundant or
Yep.
not as important.
It's the hard part of writing, right there.
Great, great. Okay, so Chris, you asked if one's microbiome plays such a crucial role in their immune system, do you think this will lead to genetically modified and manufactured holobionts? And I was wondering if I could ask you, did you have a particular purpose in mind or a particular type of organism, or was that just an open-ended question from plants to animals to us?
I was just thinking from really an open-ended, kind of open-endedly, because there's obviously a lot of different ways this could affect different organisms.
Yep.
So I think it could be applied not only to like us, but to other organisms as well, like agriculturally or something like that.
Perfect. I'm glad you mentioned that because I was going to throw that in there as well. Right. So, obviously agriculture has a history of doing artificial selection on organisms to increase crop yield, to increase how big a chicken gets, to whatever phenotype might be good for us to capitalize on. And so I think what we've appreciated is that the artificial selection is operating on nuclear genes, but it's probably operating on the microbiome as well. and in particular the hologenomic interactions between the genome and the microbiome. For example, you may be aware that chickens and other animals are fattened up by pulsing them with antibiotics. So we're changing the flora of the chickens to make them get fitter and fatter, or sort of not fitter, but fatter. So we know that we're deploying techniques already that probably are doing this kind of microbiome engineering. I think we have a much farther ways to go in learning what else the microbes can do. Maybe the scientific literacy question is how do you actually measure that? How do you differentiate microbiome selection from selection on the genome? And what would be an experiment that would allow us to test that? And just to sort of ground truth the idea that we can manufacture holobionts for good. So one of the ways you can imagine doing that is when you do artificial selection, you're propagating an organism over multiple generations. And so that organism's genetics through mutations as well as the microbes are changing in synchrony, right? The hologenome is changing in synchrony, if you will. But if you just want to know what is the microbiome doing away from the nuclear genome, you could essentially take away the genome part of the selection and only do selection on the microbiome by the following. You take an organism, and let's say you want to increase the height of a certain plant or the vegetation of a plant. Well, you could select on height or vegetation in a crop, and then once you've done your selection, you could take the microbes, let's say from the rhizosphere, the root system, purify that out, and then inoculate that microbiome collection onto a germ-free plant the next generation. And that germ-free plant would be derived from a static source of plants. And if you repeat this every generation, where you're only moving the microbiome from plant to germ-free, from plant to germ-free, and you never allow the host genome to propagate in parallel, then you could figure out to what degree does the microbiome engineer the holobiont for that particular trait versus the genome. Does that make sense?
Yeah, that makes sense.
Yeah, so I think that's going to be something that basic biologists have to ground truth. And it's also going to be something that I think is inherently applied in agriculture, and maybe even ourselves, right? Because you could consider probiotic treatments, fecal transplants, part of manufacturing our holobiont. But I think if we do this without actually ground truthing whether this can work on the microbiome, whether selection can work away from the genome on the microbiome, we don't do the science a good service. We can probably change things, but we won't know what's changing in particular. And there are good ways to do it. So we can do it like that. I think there's an area here for just some basic scientists to get involved in artificial selection experiments, and then obviously to apply that knowledge. So I think what we're seeing right now is a number of companies prop up in agriculture that are proposing to use fungi for enhancing plant crops, because people have gotten really interested in fungi that cause drought resistance in plants, or salt resistance in plants, or even too much water resistance in plants. You can imagine, spraying or somehow inoculating these fungi into plant crops that might need to grow in a warmer, hotter climate. So it could be that the microbes are really going to be an important fix for getting ourselves out of the rut of climate change and its downstream effects on agriculture. We will see. But I know people, ones that are closely allied to the symbiosis field, are looking into that, starting their own companies, etc. Okay. Mary, where are you?
Wave again, Mary. There you go.
Hey, Mary, are you a biology major or intended biology major?
Yeah.
Yeah. Did you have a particular interest that got you into biology?
Just in general, how interesting everything is, and actually symbiosis was a big part of why. I was interested in starting out with bio classes, and then I'm really certain from this class and the other science class I've taken that I'm going to go with bio.
Okay. I heard every 10 words on that, so I'll go to the question. No, I think it's a long distance issue with the computer. Yeah, so Mary, you asked, could you clarify the missing heritability issue? So one of the things that we're trying to suggest is complex traits in humans, complex diseases, and also complex traits in animals and plants are often hard to map to nuclear genes. So a certain metabolic syndrome, for example, it may have multiple causes. It may be linked to hundreds of genes. And when biologists sample human genomes and try and link variation in the genome to these particular traits, they call this a genome-wide association study. The idea is, can we find the genes? Can we even find the correct mutations in those genes that associate and perhaps are causing the phenotypes? And one of the mysteries or the missing heritability problem is we just can't find the big genes. In fact, it's really hard to find even small effect genes. And it looks like when we find genes that have a small effect on a trait, there sometimes is hundreds of them. And so it looks like the signal of what's causing these complex diseases may be very difficult to map genetically. But from a hologenomic perspective, the answer may be sitting right in our face, which is perhaps it's the microbial variation that determines that disease state more than the genetic variation, or that there's some interaction between the genetic variation and the microbial variation. And so what if the missing heritability of these complex mappings can be traced to the microbial diversity in and on you rather than the nuclear genes in your genome? Or it's some smorgasbord of both, which is more likely. But I think as we look at chronic illnesses, chronic diseases, let's take Clostridium difficile, which is this gut infection that causes year over year severe pain in the gut, severe bleeding from the gut, severe diarrhea. Some elderly people as well as children can die from it. And it's a hospital-acquired infection. It's also multi-drug resistant, so antibiotics don't work very well on it. And you ask, well, why does somebody get a Clostridium difficile infection and somebody doesn't? Or why does somebody get sick with it and somebody doesn't? And it turns out that you can predict whether somebody will get sick or not based on with high accuracy, based on their microbial diversity in their gut. That is essentially what could scale up to all types of diseases where it's been difficult to map the genetic basis of those traits. So this is a good learning situation where we can't just get fooled by personalized genomics and precision medicine driving the answers, because the answers have largely been missing. That's the missing heritability issue. And that the holobiont hologenomic perspective may be the right way to go. It may be more profound, may get to the answers quicker, and we may get therapies faster that way too. So it's sort of up to the science to drive us in this direction. There's already lots of good studies that are pointing to microbes being good indicators for whether somebody will get sick or not. And I think we will push this forward as far as we can and see where it takes us.
So Seth, let me take a break real quick here because we have about 5 minutes left. And I want to remind everyone to make sure you pick up your handouts because our speaker on Monday is Dr. Leah Ramarheim, who used to be an undergraduate student with me. And she'll be talking to you about toxoplasma. Or maybe we're forced to talk about toxoplasma by the toxoplasma. So back to you, Seth.
Okay. Elena, are you there? Hey there.
Hi.
What are you writing your term paper on?
I'm writing mine on the Tasmanian devil facial cancer.
Fascinating. Do you guys just pick your own topics?
Yeah.
Yeah. I make them talk to me about them first.
Okay, good. All right. It sounds like a potpourri of projects. You just pick them out of a hat or out of each other's brains, I guess.
Well, the parasites make them pick particular things, Seth.
Yes, right. Of course. All right, we'll wrap up with Elena's question, which is how do the genetics of a host, take humans for example, impact the hologenome? And is it more about the impact human genes have on the body? and by association, a holobiome. So let's think about this from a community perspective or an ecosystem perspective. You are a walking, functioning ecosystem, but that ecosystem is established by you being a foundation species. The host is the foundation species for which all other microbes are colonizing, right? So in effect, the foundation species will have a large effect on what colonizes that particular animal or plant, even beyond you. But this can get really host-centric really quickly, which is somewhat of a problem, because we often will resort to, and this happens in the biological literature as well, that the host is controlling which microbes colonize or not. And that is inaccurate, right? The host doesn't control anything. The host just sets up the foundation for which interactions will occur. In fact, if a host is susceptible to a particular infection, the host isn't controlling anything in that case. It's actually the bacteria that may be controlling its colonization of that particular susceptible host. And I like that nuance because I think it gets us out of this host-centric dogma. That is just part of humanity, right? Part of doing science is we start from that position. But by having a holistic framework, I think we can get out of that. So instead of thinking about your genes affecting everything, we have to think about the interactions that affect the ecosystem. And it's not you versus them, but it's actually both sets are bringing genes to the table that establish colonization. So we recently published a paper, a perspective in science on this, actually just a couple months ago. My graduate student authored this, and it was basically all about this, that we can't have this intrinsic focus on host controlling. And if a host does control something, should we assume that's all good? No, we shouldn't. Because hosts may be susceptible, if you want to take that controlling metaphor, then hosts would be controlling not only the good stuff, but also the bad stuff. And so what hosts control could also be pathogens as well as mutualists. And I think that's where the logic actually gets flawed, because we wouldn't think about a host controlling the pathogen per se, if the pathogen is winning in that particular case. Okay.
Any last thoughts, Seth?
I think that's a good perspective. I started off obviously with a history of how I got into science. I think we're talking now about you guys and how you should be thinking about science and biology. And whether you take to biology or not, whether you take to symbiosis or not, just bring the scientific literacy that you're gaining from Doc Martin's class to whatever endeavors you do, and you will have more power over your life than those that don't. And that's the true value of science and the scientific method, because we know not everybody will leave that we train as a scientist, but we can have some influence and positive influence on how you go about and think about the world, make your decisions in whatever realm they are. So thank you for the opportunity, Doc Martin. Thank you guys for your questions and comments. I can answer anything else if you guys want to get in touch afterwards. I love the art that you guys do. If you make any art and do it on some of these topics, send it my way. Appreciate it.
Wouldn't it be great to have like the letter I made up of swirling cells?
That's awesome. Yeah, or microbes, exactly.
Yeah, because that's really the truth of it, isn't it? There is no I.
Exactly.
Right. So let's give a round of applause to Dr. Seth Bordenstein.
Thanks, guys. Thank you. Appreciate it.
Thank you so much, Seth, again. And
My
I'll
pleasure.
be talking to you soon.
Okay, bud. Bye-bye.
Bye.